All right. While people are filing in, maybe I can give you just like a rundown of the plan for the day. So we have an hour and a half, and I have a bit too much probably material to present in an hour and a half because we have one constraint that this afternoon we're going to have a tutorial, like playing with actual data. So I want to get to set up the problem that we're going to solve this afternoon. So my goal is to maybe spend another half hour going over this problem of this competitive Lotka-Volterra system and then switch gears entirely and move to data. But just as a recap, yesterday we finished our preliminaries of Lotka-Volterra, and then we looked at a simple thought experiment in which we put together random species and we see what happens under some conditions. And I forgot to mention something funny, like that this experiment is named the random experiment, name invented by Yuri Barabbas. And the idea is you go to a large zoo, right? Like, for example, here is Lincoln Park Zoo in the north side of Chicago. Then you fence it off. Then you open all the cages, right? Like, and you let all these animals free. And these animals are fundamentally random animals that we picked because we like them. And so you will have the interaction between a penguin and a lion that never, appeared before in history, right? So that's why I'm saying it's like some sort of random interactions. And then the goal would be to come back after, say, 200 years and see how many species coexist, right? So this is the type of experiment that we did yesterday. And then, so we did this for highly symmetric, like system in which all the parameters were sampled from distribution were symmetric about 0. And we showed this result that either having all the species growing logistically or under this notion of strong stability of interactions, having random interactions provided the same result. And the main result was that the more species you put in at the beginning of the experiment, the more you get out, which is similar somewhat to what Sarah was showing before with this Shannon diversity. And that this also is something that you find, in fact, in some experimental settings. And then we switched gears and we were thinking about competitions, right? So experiments in which we have all the species that grow when they're rare, right? So we can take these R out. And as Sarah was saying, this is the type of thing that you want to do experimentally because you don't want to waste money growing species that then don't grow. And then we set up this problem of saying, well, imagine that these matrix of interactions is actually the sum of two matrices, right? Baseline matrix of interactions B, and then we can increase more and more the interspecific competition, right? Meaning we make the competition with the conspecific stronger and stronger. And as Sarah was mentioning just a minute ago, this is something that you find in basically all theories of coexistence in ecology. And we want to know, in particular, how much should we crank this up, right, to make the existence of an equilibrium x star, say, for this system of equation feasible, right, with all positive components. So that's the problem that we set up. And we said, well, to do this, we can actually, it's convenient to split the matrix even further, right? Instead of writing B, we write B as the sum of two matrices, one that contains the means of the columns and one that contains the remainders or the deviation from the mean, provided that alpha is positive. This matrix, because we subtracted the mean, as column summing to 0. And then we said, we call this competitive when R is positive and this M is also positive, right? So competition is the primary mode of interactions in the system. And because solving this system of equations for arbitrary matrices or for random matrices is quite difficult, we decided to study a simpler system and prove that solving the simplest system in Y, we have that Y star is positive, implies that X is positive and vice versa, right? So this is an if and only if condition, provided that these conditions are satisfied. Okay, so that's where we left yesterday. And today, the first thing I would like to show you is that indeed, we can take alpha big enough such that the feasibility of an equilibrium of these Y star or X star is guaranteed, okay? And you can think about it like in an intuitive way, but there's a slightly mathematical problem with that. So if I have these equations, imagine that I have alpha I plus C times Y is equal to 1. Then I can divide all the equations by a constant that is positive, alpha, right? And so I would get I plus 1 over alpha C y is equal to 1 over alpha 1, right? And so now it's easy to see what happens when I increase alpha larger and larger and larger. This matrix here becomes closer and closer to the identity matrix. And then I would have y is equal to 1 over alpha 1, right? So you can think that the limit of large alpha must be 1 over alpha 1. The trouble is what? That also this number becomes smaller and smaller, right? So this is intuitive, right? But it's not super easy to do mathematically. So I'm going to just do something very similar, but then we can prove a more robust result. And to do this, we just need like a Perron-Frobenius theorem. So the way we're going to do this is when we sum all the equations, we get this relationship that if I sum all the y's and then divide by, say, n over alpha, I get 1. Right? So the mean of the y's is known. Okay? So imagine that I take this thing, means also what? That, well, we can say, well, then I can take this one is 1. So I'm going to multiply this equation on the right by 1, which you can always do without changing the result, right? So I'm going to write this, and then I'm going to multiply this by alpha over n, 1 transpose y, right? Which is 1. Okay? So if I do this, then this alpha cancels with that alpha. So you get 1 over n, 1 transposed y, right, is equal to the right-hand side. Let's expand the right-hand side. I get y plus 1 over alpha cy is equal to this. And now I'm going to rearrange it by bringing these equations to the right. Okay? So then I have y is equal to 1 over n, 1 transposed y minus 1 over alpha cy. And then I'm going to gather y. Here I get 1 over n, 1 transposed minus 1 over alpha cy. Okay? Now, when you massage this in this form, everybody should go, but this is what? What is this type of equations? Right? Which I could write in general as A v is equal to lambda b. Right? It's an eigenvalue eigenvector equation, right? So this matrix has an eigenvalue of 1, right? And y is a right eigenvector for this matrix. Okay? Now, how many people are familiar with the Perron-Frobenius theorem? Some. But we just need one thing about Perron-Frobenius theorem, so I'm just going to read it here. Perron-Frobenius theorem. So Perron-Frobenius theorem is a theorem that concerns a positive matrices, or like matrices with positive coefficients or non-negative coefficients. So let's imagine for simplicity the matrix is positive, right? So all the numbers in all the entries of the matrix are positive. Then this matrix has a single positive eigenvalue, R, right? So if I plot the eigenvalues of this matrix, this is the real part of lambda, and this is the imaginary part of lambda. Then there's going to be a positive real eigenvalue rho, right? And then I can draw a circle in the complex plane such that all the other eigenvalues of the matrix are inside this circle, right? So this one is the spectral radius, right? It's the largest eigenvalues in modulus. Moreover, and more importantly for us, the eigenvector associated with this eigenvalue can be made all positive, right? You know that eigenvectors are defined up to a constant, right? So imagine you take whatever eigenvector like MATLAB or R spits out, divide by the mean, right? Now you made it all positive, okay? So this is guarantees the existence of this eigenvalue and of a positive eigenvector, okay? This matrix, is it all positive? Depends on the value of alpha, right? But you can imagine that if I can pick alpha such that 1 over n is larger than the minimum of Cij over alpha, right? So like the most negative number, right? Like, or the most, or the maximum. Say the maximum because we have a minus here. Maximum over I and j of this number, right? Now this matrix has all positive entries, right? It also has, because when I sum, these column sums are 0, so the sum is actually 1 for each column, which means that this is a column stochastic matrix which has 1 as an eigenvalue. And because of Perron-Frobenius theorem, this is the spectral radius. And therefore, y is positive. Okay, so you don't need to go to infinity, right? You just need to go to some number that is between 0 and this number here. Does the logic of this make sense, right? So we can choose alpha such that this whole thing is positive. It has 1 as a spectral radius because all of the column sums to 1, and because of Perron-Frobenius theorem, and then y is guaranteed to be positive. Yes? If this is confusing, I can repeat it. All right. Right, so that I can pick an alpha, say that alpha infinity is an alpha large enough such that Cij over alpha, or like 1 over n minus Cij alpha infinity is greater than 0. Then I've made this matrix all positive, so it's guaranteed to have an eigenvalue, right? That is a positive, right? This rho. And because all of these column sums to 1, right? That has to be the spectral radius, has to be 1, which is exactly the eigenvalue that we have. Where is it? Here, right? This is lambda equals to 1, right? So that has to be the spectral radius, and therefore the eigenvector associated with the spectral radius because of Perron-Frobenius has to be positive. Yes. Here, what I just wanted to show you is that the intuition that we can pick an alpha big enough such that feasibility is guaranteed is true. And we don't need to really go out of our way, like, and put the alpha is equal to 10 trillions, right? Like, to get there, we can just like, this one is already sufficient to guarantee feasibility, right? So one way to think about this is like, if I were to say, imagine that I want to plot what is the probability of feasibility when this matrix is somewhat random, right? Or, so imagine that we have some sort of a random matrix, right? So then it makes sense to think about, if I do construct 10,000 matrices, right? And I count how many times do I have a feasible equilibrium, right? That could be an approximation of a probability. So imagine that here we're saying, what is the probability that x is all greater than 0 on the y-axis? And then here we have alpha. What I just showed you is that eventually there must be some sort of critical value, right? Alpha infinity, and there must be some value that is at most this, such that the probability is 1. Okay? All right. In fact, this is the object I would like to derive next, is what is the probability of having a feasible fixed point when this matrix is random, right? So when we sample the entries of B from some random distribution. Okay? And to do that, we're going to use some random matrix theory, but especially we're going to think about the case in which this model is really unstructured. And by that, I mean that there is no, in the model definition, right, when we sample this parameter, we don't have any species that is peculiar or special, right? Like that all the species are statistically the same species, right? So what I mean by that is, imagine that you're sampling all the entries of, for example, B, right? Imagine that we're saying Bij is taken, are all independent, identically distributed from some distribution Q, such that the mean of Q is positive, right? Because we want to have some sort of a competitive system, and then the variance of Q is some number, right? Like it's equal to sigma squared, right? Well, for example, that would be. Now, if I relabel all the species, right? Before I had 1, 2, 3, and now I have 3, 1, 2. The model definition is the same, right? So these species are all somewhat equivalent to each other, at least from a statistical standpoint, right? Of course, when for any iteration of this model, the exact value of each species will be different, right? But if I look over many, many simulations, I would have that the mean of the species 1 is equal to the mean of the species 2, etc. Okay, and that's exactly what I mean by a statistical equivalent. I mean that the statistics on these species, for example, the expectation for the value of species 1 or species I is equal to that of species J, right? And then the variance of the component I of the vector Y is equal to the variance of that of j, and then the covariance between, say, yi and yj is equal to the covariance between yi and, for example, yk, right? This is what I mean by statistically. I don't have an a priori reason to say, oh no, species 3 is different from the rest. Then, like, just as a reminder, we're going to do some small manipulation of these kind of quantities, right? Expectations, variances, and covariances. So I wanted just to remind you of the one possible definition of the variance of a random variable, let's say x, is like the expectation of x squared. And maybe I should use only round parentheses. Expectation of x squared minus the expectation of x squared. Okay? So this is true in general, but this means that I can find 3 different expressions, right? If I want to solve for this, or solve for this, I can't, right? And in particular, we're going to use the expectation of x squared has to be equal to the variance of x plus the expectation of x squared, right? So you just switch these 2 terms. And then the other thing that we will need is like the covariance between x and y is defined as the expectation of x times y minus the product of the expectations, right? X, y, right? And again, if I want to write, for example, the expectation of x and y, right, I can write the covariance plus this object, okay? This is all that you need to remember from your random variables. And as I showed before, we have that the sum of all the y's is fixed. And so I want to show you that when the sum of all the y's is fixed, as in this case, and we have statistically equivalent species, we can only have one covariance matrix, irrespective of how complicated your model of statistically identical species are, because we could think of a simple case like the one I showed you, like where we sample all the entries from one distribution. But we could sample all the entries in pairs, right? We sample Aij and Aj from a bivariate distribution, or we could sample it in more complicated, like little pieces, like with 3 things at a time, or higher order correlations. You can do anything you want, as long as when I permute the labels of the species, I have the same model definition. These are statistically equivalent species. So what we're going to derive, so right now we've derived results that hold for any b. Now we're looking at random b's. And so these will hold for any model, random model, such that species are statistically equivalent. And our starting point is what we already wrote a few times, right? That like the sum of all the y's divided by n, so this is the mean of all the y's, is equal to 1 over alpha, okay? If this is true, then this must also be true, right? If I square this and I square this, they also have to be the same, thank you very much. Also because it's positive. And so I could write this, for example, as the sum of the yi squared is equal to n squared over alpha squared, right? Because I bring this thing on the other side, okay? And now I'm gonna start writing these things in these terms, right? So first I split the sum of the yi squared as the sum, for example, over j of yj squared plus the sum over I, sum over j different from I, right? So this would be the diagonal terms, right? And then of yi and yj. This has also to be equal to the right-hand side. Okay. Now, what is this value, right? This value, there's n values, and then there is some sort of expectation for x, for yi squared, right? So I can write this as n times the expectation of yi or yj squared, okay? Because this is a sum. And then I can write this. How many elements do we have? N times n minus 1, right? Because we took out the diagonal, so n times n minus 1. And what is this going to be? The expectation of yi, yj still equal to n squared over alpha. Okay, now I can get rid of, for example, 1n here. And then I can write this expectation as the sum of the variance and the expectation of x squared. This is for one component, because y I call the whole vector. So I just put a generic, just to say it's one coefficient, it's not the whole vector. Okay, so we can write this as then the variance of yi plus the expectation of yi squared, right? That's this term. And here we can write it as plus n minus 1 times the covariance of yi yj plus the expectation of yi expectation of yj. And this is equal to n over alpha. So far, so good. Now, if these pieces are statistically equivalent, what is this? Exactly. Expectation of yi squared. Okay, now let's count how many expectation yi squared do we have. We have 1 here, n minus 1 here, so we have n times the expectation of a yi squared. What is the expectation of yi squared? Here is the expectation of yi squared. It's going to be 1 over alpha squared, right? So, and here we have, sorry, this is squared, right? So then all these terms and these cancel. Okay, everybody follow this trick? Good. So now we have that the variance of yi plus n minus 1 times the covariance of yi yj, which is the same for all pairs of yi yj because of this statistical equivalence, is equal to 0. Okay. Now, if I divide everything by the variance, what do I get? The correlation, right? So, because again, the standard deviation of yi and also yj are the same. So then we get the correlation between yi and yj is equal to negative 1 over n minus 1 for all I and j, right? So in general, what do we know about this system? Without even knowing how we build the system, but just knowing that these pieces are statistically equivalent, we know the mean, we know the correlation. The only thing that we don't know, and the only thing that will change under different definition of these models, is the variance, right? So if we have the variance, we have the whole a picture of both the mean and the variance covariance matrix. Okay? This would be when I do like that's a very good question. Like, what does it mean to have a correlation? It means that when this thing tends to be high, the other thing tends to be low. This could be due to a number of reasons. For example, things compete, right? So when I have a lot of this, I have less of that. But it could also be that the correlation between the equilibrium values is due to, for example, in predator-prey system, right? Like the simplest case that Volterra was doing, like, this is my prey, this is my predator, right? And I have an equilibrium. And then I change fisheries, right? Like a fishery effort. For example, like Umberto d'Ancona was looking when they closed the fisheries, right? This thing will move in this direction, right? So if I were to take this snapshot at different times, right, I would get a negative correlation in this case. This doesn't mean anything about the species per se, but it tells you about how the environment has changed, right? So it's not directly interpretable, but what I'm saying is that we want to know what is this probability of feasibility, right? What does it mean to have something feasible? It means all the y's are greater than 0. And we know that because this is some sort of random system, then these y's will have a distribution. And what I'm telling you is, of the distribution, without having done too much math, we know the mean and we know the correlation. The only thing we don't know is the variance. Okay, fantastic. So now we want to compute this variance, right? If we could compute this variance, we would be in business. Just like before we get there, I just wanted to show you how would we close this problem of computing what is the probability of feasibility if we knew the variance, right? So we're saying this a random variable y, right? It has y is taken from some distribution such that the mean is the same for all of them, right? It's going to be a 1 over n, 1 over alpha, right? 1 over alpha, 1. This is my mean. And then there's a covariance matrix that is sigma square of y, which we don't know what it is. And then my covariance matrix is 1 on the diagonal, negative 1 over n minus 1 on the off diagonal. Okay, that is true for all these models with the statistical equivalent species. And now you can make an argument for saying this has to be a normal distribution, right? And I'm going to spare you the details, but you find them in the paper of why do you want to do this. But then bear with me for a second and say that this is indeed a normal distribution. Now I want to know what is the probability that all the components of y are positive, right? What should I do? I should just integrate, right? I can integrate between 0 and infinity, etc., for all the different species. This density function, right, of the normal distribution, that gives me what is the density of y in dy1, dy2, dyn. Right? These are called orthan probabilities. This would be the probability that x is greater than 0. Okay? So these are called orthan probabilities. In the case where you have such a simple covariance matrix, because you can see that this covariance matrix is a rank 1 perturbation of the identity matrix, you can turn these multiple integrals into a single integral. The problem is only that when the correlation is negative, this single integral is in the complex plane rather than being, plain vanilla integral. So that's one option. The other option would be we have this distribution. We could look at the distribution of the minimum, right? I would say, imagine that I always put the species with the lowest abundance, call it y1, right? The minimum of all these numbers. Then you can use a similar technique that is based on order statistics, right? Which we're going to see actually tomorrow, like something about order statistics. I take numbers and then I order them and I want to know what is the distribution of the minimum. You can write that down. And so then would be, if the minimum is larger than 0, then it means that all the other ones that are larger than the minimum are also larger than 0. But if we care about like the case in which there's very many species, what is minus 1 over n minus 1? It is 0. Who cares? We're just going to neglect this term altogether, right? Because we're in the space of there's many, many species. And therefore, this calculation becomes much simpler, because if this were to be 0, all of these y are non-correlated, right? They're IID random variables. And so let's think about that case for a second, in which we're assuming that the correlation is in fact basically 0 or negligible at least. So now we want to know what is the probability that yi is greater than 0, right? And then we're going to raise this probability to the nth power to get all of them. Okay, so the probability of this one is fundamentally the probability that a random variable with mean 1 over alpha and certain variance is greater than 0, right, which is a normal distribution. Then we could say, well, I want yi greater than 0. Then I can subtract the mean from both sides, right? So let's say yi minus 1 over alpha is greater than minus 1 over alpha. Call this thing, I don't know, zi, right? So now we have the probability of this. And then we could divide both sides by the standard deviation of y. I call this thing, give me a name, theta I, sigma y. And because the normal distribution, which now has mean 0, is symmetric about the mean, this is also the probability that is greater than, right? It's the same. So we just need to take what is the probability, the cumulative function, right, of the normal distribution, evaluated at this point, right? 1 over alpha sigma y raise it to the nth power, because all of the species have to be positive, and this is my probability of feasibility. Okay, so that's the thing, which tells you, if we were only able to compute this one number, right, we just need one number, then we would have a scheme to approximate this probability of having a feasible solution. Okay? No, because I removed the mean. This is symmetric about 0. Yeah, that's why I removed the mean, to make it 0, such that I don't have to deal with the negative. Okay? So the question is, how do we compute these variance? And that is like a tricky problem. So I'm going to show you one approach. Like if you read the papers, there's 3 different approaches, right? That tells you about the desperation of trying to compute this number. We tried many, many, many things. In the end, 3 of them, we were able to carry them to the end. Some, use like random matrix theory. One uses random matrix theory. I'm going to skip over that. One is like requires some more heroic calculation, but it's very easy to understand. So I'm going to do that. And there's another method that uses fancier mathematics, like the theory of resolvents, which turns out to be the one that works the best. But I don't have really time for any of it. So I'm just going to show you like the gist of how can you compute this variance in a simple way. And then I show you just results of simulation of, if you do a more heroic calculation, what would you get? Okay? Good. So to do this heroic calculation, what we're going to exploit is what is called the Newman series, which is another way to express an inverse. You remember yesterday we were looking at Sherman-Morrison that says, if you have one inverse, now like you can get another inverse for almost for free. This is a similar result in which it says, under certain conditions, so imagine that I have a matrix, let's call it, I think I call it T in the notes, right? So imagine that I want to solve this system of equations, right? And what I need to find is the inverse of I plus T. So it turns out that if the spectral radius of T, right, exactly the same type of thing we were doing before for the Perron-Frobenius theorem is less than 1, so this has larger eigenvalues than that, then you can express I plus T inverse as an infinite sum between k equal to 0 and infinity of, let's put negative here because it looks prettier, of T to the k. Okay, so this is called the Newman series. Yes?
But somehow, if we don't have large enough numbers of species, we get covariances in their abundances.
Yes.
Despite having random interactions.
Yes. If you have some numbers and their mean is fixed, they will always be negatively covariant. There is no way around that, right? Because if something goes up, something else has to come down, because otherwise, how can I maintain the same sum? Does this make sense?
Yep. Okay.
Yeah. So this is true. That's exactly how you get, for example, the Dirichlet distribution is exactly like in this way.
If theta is distributed symmetrically about the mean, then I think it should be less than 1 by sigma, alpha sigma y.
Oh, yes. Sorry. That's lower or equal. Right. That's why I want to use the CDF. Yeah. Sorry. Thank you. All right. So what is the idea? We have a very similar problem, right? We write 1 plus 1 over alpha c y is equal to 1 over alpha 1. And now we're going to express the inverse here as an infinite series, right? So we will get y is equal to 1 over alpha, sum between k equals to 0 and infinity of minus 1 over alpha c to the k 1. Okay? Now that you have this, you can say, well, now I can approximate y by taking the first term would be c to the 0, which is the identity matrix. The second term would be c. The third term would be c squared, etc., etc. Right? And every time you add one term, you add to the heroic calculations. So I'm going to just, go ahead. What is what? Oh, this is a generic matrix. This is just to explain the Newman series. It's just that this is the notation I have in the notes, so I use that for simplicity. So I'm just going to show you the calculation because otherwise we're going to run out of time. So this is the Newman series, right? And so we do this approximation. And so you can take the first term, right? You would get just the identity matrix. The second terms, you would get this row sum of C, row sum of C squared, et cetera, et cetera. You keep going and going, right? Once you have approximated Y, you can compute its variance very easily. Okay, so let's see if we wanted to take only the first two terms, right? You do a little bit of algebra, and you end up with like an estimate for the variance of Y would be this horrible object. This horrible object, you know that this is a random matrix to which we have subtracted the means. So you express that in this way, and you get this other calculation. And then doing some algebra of random variables, you can find expectation of all the pieces. And then you end up with something that looks like this, right? So the expectation for this variance is approximately n-1 over a alpha to the fourth. Okay, let's see how it works. So what you can do is you start at different alphas and you build many matrices, and you check what is the probability that this is positive, right? And what you find is some sort of sigmoidal curve, right? Here I have, for example, for 250 species and 10,000 draws of this, right? The dashed line is the simulations. And this would be the approximation that you would get using this normal distribution for this heroic calculation that we did, which is kind of disappointing, right? Because it's not very good. So then you say, well, then I can take more terms of the Newman series, and now you get an even more heroic calculation, which I'm going to spare you altogether, or you use these fancier methods, and now you can get this slight change, right? Before we had just alpha to the fourth here. Now we subtract n-1 alpha to the square. After having done all this calculation, that's what you get. And then you go from the red line to the green line on the slides. So this one is a very good approximation.
For truncating the Y series on to two terms, you must have some conditions on the terms, right?
You just need this one, that like this alpha has to be larger than the spectral radius of C. And you can make an argument for why this is the only case in which you would have feasibility, which I also spared you. Okay, so bottom line is, with some heroic calculation or using some more clever methods, you can get a very good approximation of this that looks like a very difficult problem. Like what we just asked is, if I have a random system of equations, of linear equations, and I can tune the diagonal, right, however much I want, what is the probability that this system of linear equations that are random has a positive solution? And this is the answer, okay? Now, this we dealt with feasibility. What is the other thing that we've dealt with yesterday and also on Monday? Stability, right? And in particular, we were saying, oh, if this matrix has some sort of special property, right? The symmetric part of this matrix is a negative definite, or in this case, because we put a minus positive definite, then feasibility implies stability, okay? So where in these alphas would be an alpha? So we want to ask, is it true that alpha I plus B, if I take the Hermitian part, is positive definite, right? What is this object? This is 1/2 alpha I plus B plus 1 alpha I plus B transposed, right? Which is alpha I plus 1/2 B plus B transposed, which is, in fact, the H of B, right? So you have a matrix which has some eigenvalues, right? Imagine in this case, H of B will have only real eigenvalues because it's a symmetric matrix. And then you add a positive constant to all the eigenvalues. What happens to the eigenvalues? They all shift to the right of exactly alpha, right? So you can imagine that there must be an alpha big enough such that I crossed all the eigenvalues into the positive line, right? Does this make sense? So what is this critical value of alpha? Turns out you can use random matrix theory to compute it very easily. In fact, like the distribution of eigenvalues of a random matrix that is symmetric follows what is called the Wigner semicircle law. This is one of the first results in random matrix theory. Oh yeah, it should be negative definite, but because in all my equations I have a minus ax, right? We were saying a has to be negative definite, then negative a has to be positive definite. Okay, so it's a potato, right? You can always switch it by switching a sign. But here is, for example, the spectrum, right? Like how frequently do you get eigenvalues between one number and another, right? When you do a large random matrix with the symmetric and identically distributed entries. And the red line is what is called the Wigner's law, right? Like Wigner's semicircle law. So you can compute this number quite easily. And unfortunately, we don't have time to compute it. But what I want to show you is where is this critical alpha that would make this thing positive definite. Where is it located? It's located here, right? It's the blue line, which tells you what? That what is the probability of feasibility when we just cross this critical alpha to make it stable? Not zero, but very close to zero, right? As soon as we have a reasonable probability of having feasibility, we get stability for free, right? Which means in turn what? What were we saying? That this feasibility is necessary and yet not sufficient for coexistence. So if I do this experiment at different alphas and I find something feasible, almost invariably, unless I'm very lucky and I get something feasible when the probability is 1/2 to the n, then it's going to be automatically stable, which means that are we going to observe cycles or chaos or other things in this system? We will not, right? Because we're already crossed into the area where all that happens is that we go to some sort of saturated equilibrium, right? So like this calculation of probability can tell you something about the dynamics of this system. Okay, I'm sorry that I had to cut this a little short. Fortunately, like I spent a lot of time doing these notes, so you can read the notes, but I also put some papers that you might be interested in. So this Servan et al. Is like the paper on the first part, like this random zoo. What happens when we put a species at random, etc. This is the experiment with the pitcher plant that I showed you the results of. So if you want to read the results, this one we already discussed, chaos and cycles in competitive systems. And then this approximation of the feasibility has a long history. And so there's many attempts at getting to this probability. I put here like 2 representative papers out of many that are available. And then the last one is a preprint, which is exactly the material that I presented. And of course, a lot more. Like this preprint is as long as Anna Karenina. It took me like the whole summer to do this, right? So this would conclude this part of random systems. So while I raise the board, if you have questions, I would be happy to hear.
The fact that you have a critical alpha beyond which you see feasibility, is it similar to the modern coexistence theory that was talked about in the previous lecture?
Right. Yeah, like as I said, like this idea that you need sufficiently strong self-regulation or interspecific competition to have coexistence, so you have to have different enough niches, is present in basically almost any theory of ecology on coexistence, right? So you will always find this thing that, yes, having a strong competition with your conspecific such that you don't blow out to infinity, it's typically a good thing for coexistence.
Can you also do this for, let's say, motifs or strongly connected communities? Like if you code screen them and say that they are mostly reinforcing themselves?
Yes, but the trouble there is, like, imagine that I have, I don't know, a lake, right? And this lake is stratified such that there is like a community in the pelagic part, like the upper part of the lake, and one in the benthic part, right? So you could think, well, these things will interact mostly pelagic with pelagic and benthic with benthic, and I will have maybe some exchanges, but not so many. Fine, right? You can do the same calculation. But the thing that you lose is the statistical equivalence. Right now, belonging to the pelagic or to the benthic matters, right? Because you will have different categories. So you have to do basically the same calculation for a block structured matrix, which can be done and has been done for stability, for example, by Jacopo here, right? Jacopo, how would you describe your experience? Traumatic or non-traumatic? Yeah, so the calculations get a little hairy, I would say, but it is possible in principle, and I think like once you start opening the bottle of this calculation can be done for this case, then there's somebody smarter than I am that can do it for another case. And I'm happy to just sit down, relax, and eat my popcorns while people do these other calculations.
In network theory, we generally have a set of random networks and then small world networks. So here we considered random interactions. Would there be such an equivalence here?
Right, that is the similar case, right? Like imagine that you have some sort of power law degree distribution, then the species are not equivalent, right? Because there's one parameter, the degree, that would discriminate between the species. Again, I'm sure that the calculation is possible. Do I want to do it? Absolutely not. I want to present it to people such that somebody says, I want to do this calculation. Be my guest and do the calculation yourself. All right. So in the interest of time, we're going to switch gears completely. So I presented something highly, proudly theoretical, I would say, right, where we talked about infinitely many species, picked at random, etc. Now we're going to go back to Earth for a moment. I rarely do this, but sometimes even I use data, I can admit it. And we're going to look at experimental data, very much of the kind that both Daniel and Sarah have presented. And so the idea is that we have a species pool, right? If we have a bunch of species, for example, the 4 species of the metal oil that we've seen before, or some other species from the gut microbiome, or could be also plant, protozoans, whatever you prefer. And now we're going to do experiments in which we co-culture different combinations of these species. My favorite number is what? 2 to the n. How many possible combinations there are of I put this or not? 2 to the n, right? It's always the same number. Okay, so these experiments have been conducted in the history of ecology with plants, famously these biodiversity ecosystem functioning experiments where you have plots and you put 2 plants, 3 plants with a legume, without a legume, and you see what happens. With protozoans, for example, Gauss, right? I was doing these type of things with protozoans back in the day. And with bacteria, you just see some experiments with bacteria, so you know that this exists. In many cases, people don't want to spend the time and money that Sarah spent to do all this beautiful time series, and so that we could muse on what is going to happen in the system for half an hour. They have either limited means or like limited time, so they only look at what happens at the end. I put A and B, and at the end I wait for long enough, and then I sample what happens at the end, right? And this is what, because we can only do this kind of experiments with a reasonable number of species, right? Because as I said, the number of possible experiments we can do grows exponentially with the number of species, right? So if you have 2 to the n minus 1, because we don't do the experiment where we put nothing and see what happens, because Spallanzani was right and nothing will happen. But you know, if I have 10 species, I have 1,000 experiments. If I have 20 species, I have 1,000,000 experiments. So that's not really great. Okay, so imagine that this is the setting. We put some species together. We grow them for a sufficiently long time such that they do what they have to do. And then we observe the system at the end of the experiment. And as I said, many people did this. So I'm going to show you some results that were published this year, not by me by any means, in PNAS, where these Ishizawa and colleagues did some sort of heroic experimentation. So they took 7 strains of bacteria that are associated with duckweed. Duckweed is like the water lentil, right? Like little plant that floats on the top of like ponds typically. And then they put them in vials and infected these plants with different combinations of these 7 bacteria. How many combinations do you have? 127, right? Because we don't do the one where we put nothing. Okay, so they keep them in this little flask for 10 days, and then at the end of the experiment, they plated the microbial community in 7 different plates, each one of which had antibiotics that would kill all the bacterial species but one, so that then they could count how much there was in each. And the data is really incredible because there are 692 experiments, right? So they did each one of the 127 in several replicates, not exactly all equally replicated, but you know, an impressive system. So because we don't want to look at 127 plots at the same time and spend like a lot of time computing this afternoon when we're going to do this computationally, we're just going to look at some experiments in which there were 3 species. So we look at all the possible combination of 3 species, which is 7 combinations, which we can visualize quite easily, right? So this is an example of the data. So you can see that here there's on top 2 experiments where they put only species 3. Then here there's 2 experiments where they put only species 2, or at least only species 2 survived at the end. Here there is an experiment where both species 2 and 3 survived at the end. And this one where all the different species survived, okay? So how can we describe this kind of data, right? We have for sure, which species are we talking about? Are we talking about species 1, species 2, or species 3, right? So let's call this, I put a tilde to say experimental, right? So this is, I'm talking about species 1, species 2, or species 3. But then also, like, which context are we talking about species 1? Are we talking about species 3, for example, growing by itself, or species 3 growing with also 2? Right? So we could say, we can label this number by a community. Right? So imagine that this community says, this is species 1 when grown with species 3, or this is species 1 when grown with species 2. And then we have, so these 2 samples, for example, would be the first kind of community, and this is species 3 in the first kind of community. But then we also have replicates, right? So we can do the same experiment multiple times, and we will get, as things go, somewhat different results. So let's put also...
Stefano, a quick question. I mean, maybe you said it, but what are they doing to reach this steady state?
They wait for 10 days.
10 days and nothing, no, just add it to the duckweed and wait.
They don't even know if it's a steady state. They just do it for 10 days.
I understand.
And Stefano, related question. So this community indexing that you're doing, are we thinking about what you put in initially, or
no, the final? What did you observe? Right,
okay.
It's even blind. Maybe here they started with all the species, but they ended up with
right.
Species.
Okay.
So we're only looking at the final point. Okay, so then we have, this is a way to index all this data, right? So each one of these numbers can be written in this way. Do we agree with that? Okay, so now we're going to build a simple statistical framework, and I argue the simplest possible statistical framework we can build. So what is the first assumption that we can make that is a strong assumption, but that makes things a lot easier? We have several replicates. What do we believe? Will they all go in their own merry ways, or will they all go to the same part? Right? So if we think they will, if I do the same experiment twice or 3 times, I get the same result, then I could say, well, then these numbers are sampled from a distribution, right? Imagine a distribution Q that is a generic distribution that has a true mean that does not depend on the replicate, right? That depends only on the community. So the typical value for species I in community K should be this number. Okay, so if I do many experiments, right, I have, this is like the value of xi in community K replicate R plotted across all the replicates. I'm saying there is some sort of mean. Okay. And then there would be some other parameters, right? For example, if you want to say this is a distribution that is controlled by 2 parameters, 3 parameters, then you could put like this. There are parameters for species I in community K. And again, these parameters do not depend on the replicate. Okay. So in practice, what kind of assumption are we making when we do this simple model? They're saying these then are independent, identically distributed samples. We're saying if I do the experiment many times, I go in about the same place. When I put A and B together, I don't have A is like very high and B low sometimes, or B is high and A is very low other times, right? They always go to about the same point. Okay. This is to say there is no true multistability. And by true multistability, I'm saying this is like X1 and X2, and sometimes I go here, and sometimes I go there. This is what I would call true multistability. What we are saying is, if I have this system, I can go here, here, or here, right? There's no way to have both of them coexisting at completely different places. Okay, they can change in composition because then this changes the community, then we're fine. Okay. So this is a strong assumption, but I think it's one that every experimentalist would like to have, that if I do the same thing 10 times, I get about the same result. And if I don't, I worry about what am I doing wrong. Okay. Now, this model has how many parameters? A lot, right? We have one parameter here for each species in each community, and then we have ancillary parameters for each species in each community. So we will never be able to fit this model ever, right? So let's make a second set of assumptions that we're going to make, again, very strong assumptions. And what we can do is like to try to reduce the number of parameters is to connect the values of I in different communities with each other, right? And what I mean by this is to say, imagine that we want to model xi of k, right, which is the value that I reaches typically when it's growing in community k. And this is going to be equal to some number. This number we could think of when I is growing by itself, it reaches a carrying capacity of some sort. That's exactly like what Daniel showed you like yesterday, the day before. And then we could say, well, and this number is modified by all the other species that are not I, that are in community K, right? All the other species which you're growing with, according to some parameter. Xj of K. Okay? This is saying what? This is something Sarah talked about yesterday. What we're saying here is the context does not matter. The effect of J on I is the same, irrespective of the community, right? There's no K here, right? We're saying there's a fixed effect of species I on J or J on I, and that's what it is, okay? Moreover, this effect is just like proportional to the abundance of J directly. So this is a linear model. Is nature working this way? Probably not, right? But this reduces the number of parameters quite a lot, because before we had all these parameters and now we have 1 and then n times n minus 1 here, right? So we have n squared parameters. Okay, and we have many more experiments than n squared parameters. Okay, so what we're doing here is, again, a lot of assumptions on how do we relate the density of I in a certain experiment with the density of I in another experiment. Okay, now this is, we set up as a very simple statistical model, but what I want to show you is what is this? Our friend Lotka-Volterra, of course, otherwise would not be part of this series of lecture on Lotka-Volterra. How do we get Lotka-Volterra from here? Well, we can, for example, divide both sides by alpha I, which we expect to be positive, right? Because we're saying things that can grow by themselves. So we get 1 over alpha I, xi of k. And then we bring all these other coefficients to the left-hand side, plus sum over j, different from I, j in k, of beta ij over alpha I times xj of k is equal to 1. Okay? Now I call this thing bii and these things bij, right? And now what I wrote is exactly the same thing that we did for Lotka-Volterra. That says b taken only the submatrix for k times x of k is equal to 1. Okay? So another way to put the statistical framework is to say, for the means of the distributions that we've seen, is there a Lotka-Volterra model whose equilibrium structure that is defined by this matrix B, as we've seen before, it encodes all the 2 to the n possible equilibria? Is there one that is sufficiently close to my means, right? If there is, then we can do all sorts of magic with this, right? Because if we have all these parameters, not only we can fit our data that we have gathered fairly well, but we can infer what's going to happen for experiments we didn't do, right? Because now we have all of the possible equilibrium, not only the ones that we use to fit the data. Okay? So this is, what I'm saying is that this is equivalent to the structure of equilibria of Lotka-Volterra models. And I just wanted to show you one thing that people rarely think about. And it is, imagine that I build a matrix where I encode all the possible 2 to the n equilibria of the Lotka-Volterra model when they're feasible. Imagine here there's a case in which each species can grow by itself. These two can also grow in these two pairs, right? And maybe not the third pair, and then there's an equilibrium with all the 3 species present. Okay. Now, if I take this matrix, right, which is species and the different equilibria, right? That's why I called it E as an equilibria. What is the relationship between this and the matrix of interactions? Right? If I take, if I have these equations, right, and now I take a matrix of all ones and subtract E B transpose, okay? So what am I gonna compute is the inner side of these parentheses, right? We know that, for example, B X star is equal to 1 for all the species that are present, right? And then we know also that the growth rate, the intrinsic growth rate or invasion growth rate of these other species when they're absent is computed in the same way by putting this at X star and seeing whether they can grow or not. So what I get here is I get 0 in red. This is the 0 for the inner part of the parentheses, right? For example, when only species 1 is present, this red 0 means this species is at the equilibrium. What do these other numbers represent? They represent the invasion growth rate of 2 when trying to invade the system with only 1. So, right, so a positive number means this is an invisible equilibrium. It's not a saturated equilibrium, right? And then you can see here, it's also invisible by the other species. Here, it's also invisible by one of the species. These two equilibria instead are saturated, right? We have that when I have these two species together and I try to put the third species, it dies. Okay, so this is a relationship between this matrix with all the equilibria of the Lotka-Volterra and the matrix of interactions. And if there is a relationship, it means what? That from one I can learn the other, and you know how to do it already, right? If I have this system, I just invert this matrix and I can get X star. But more importantly, for our reason, because we've observed equilibria and we want to find the matrix, we can go backward, right? We can take this kind of data and try to infer what B is. Okay, so we can learn. And what are we doing by doing this? You remember that Sarah showed you, okay, what would be the sum? Oh, you have a question.
Here, if then like there is 2, if it is positive, then it can invade, and if it is 0, does it like imply coexistence?
It would be neutral, right? Like, so if I put it at very low abundance initially, it kind of hovers there. And then depending on the exact condition, because this is an approximation that you get by saying it's very small. So then, you know, depending on what happens when it's a little larger, right, you would get an extra term. This would either go to 0 or invade. Okay. So it's inconclusive, I would say, a 0. So you remember that Sarah was showing you, like, I do all the pairwise compared to the monocultures, and I can kind of get a sign of, like, is this effect positive or negative, right? And she was also saying, wouldn't that be nice to do the same thing when I have also the triplets, information on the species growing 4 at a time, 5 at a time, 15 at a time, right? And this shows you a very principled way to do it, right? I'm trying to find a solution that fits all of the observation that I had. Okay, so what I wanted to show you is how do I find for example, a row of the matrix B, right? Given the experimental data, right? So imagine that this is my experimental data, and this is not experimental. This is actually Lotka-Volterra, right? And imagine that I take this matrix E, and I select all the equilibria where species 1 is present. In this case, I have 3 of them, okay? Now, each one of these must abide by what rule? That if I take this equilibrium and multiply it by B, right, I have to get a 1, right, for all of these species. Okay, so for example, if I take this thing and I multiply by the row, the first row of B, right, that I use to simulate this data, then I have this matrix times this is exactly 1. Okay, this is this equation, right, that we're saying here, right? So if I take row I of B times X of K, right? But here I take only the K sub species, right? This has to be equal to 1. This is exactly what I'm doing there. Okay? But then it means that if I have this matrix and I invert it, right? I bring it on the other side, what I find is the row of B. Right? So what would be the row of B? Would be the inverse of this matrix. Take the row sum of that. This is the coefficient in B. Okay, it requires a little bit of algebra, but you can do it. Now, in this case, it was chosen because this matrix of equilibria, there's only 3 equilibria in which species 1 is present, and there's only 3 coefficients in B, so it works by inversion. What happens if you have more equations than you have coefficients? Right? For example, if we look at the same experiment between quotes, and we look at species 2, so we take E2 is like the matrix with all the equilibria in which 2 shows up, and now there's 4. Right? So we cannot invert a rectangular matrix. But what you can do is exactly the same thing that you would do for, say, linear regression. Right? If I have AX is equal to 1, or whatever you want, AX is equal to B, and we have that A is a rectangular. Okay? This system has no solution, right? But what do you do in linear regression? You say, well, I multiply both sides by A transposed. Now, A transpose A is a square matrix. And if this is a very large system, it's invertible. Then I invert both, like I invert this on both sides. So I get X is equal to A transposed A inverse A transpose B. So this matrix is called the Moore-Penrose pseudo-inverse, and that's exactly how you solve for linear regressions. This thing has a fantastic property that minimizes the sum of square deviations between what you predict X to be and what you have observed. Okay, so this is what you're saying is, I have point, and I'm trying to draw the baseline, and the baseline means like that if I take all these deviations from the line, sum them, once I square them, this matrix is guaranteed to minimize that sum of squares. Okay, so that's one way to recover. Of course, if these data were generated by a simulation, you would obtain exactly the same result if you were to ditch one row, right? Because these things are redundant, right? It's a rank deficient. But in real data, this could be useful. Okay, so what we want to do is to say, okay, imagine that we have experimental data, and that we have B, we have inferred all the coefficients of B, Bij, from this experimental data. And now I want to predict what's going to happen for an experiment I didn't do. Okay, so now you're in the space in which we have found B. We haven't really found B yet. We're going to find it in a minute. But like, imagine that we're there. And now what we want to know is like, I don't know, I did all the experiments with 2 or 3 species, and now I want to know what happens when I do the experiment with 4 species before I do the experiment. Okay, how am I going to do that? Right, so we have our matrix B, and then we want to figure out what happens when we get a certain community, right? So I want to know what happens if I have all the species together, or whatever combination of species you want. So you're saying, well, if this matrix B is a good description of my system, then I would have that B of K, right? Like the B in which we take only the rows and columns belonging to K, times the unknown X of K, which is what I want to predict, is equal to 1. And so I invert this matrix, and I get X of K is equal to B of K inverse times 1, right? The row sum of the inverse. Now, there is 2 options. First option is these numbers are all positive. Then what we would conclude is, yes, if you do this experiment, my bet is these species will coexist together, and they will coexist exactly as these values. Okay? If any of these numbers are negative, on the other hand, my bet would be these species will not coexist with each other, right? Because if I apply the same rules that I've used to build this matrix B to this new community, I would find that there's no way that they can have a point in the middle of the positive order. Right? So once we have B, we can do these out of fit or out of sample experiments and validate our approach in this way. The way we will do it this afternoon is you take a bunch of data, remove all the experiments in which there is a certain community, find the matrix B without this data, predict what's going to happen to the data that you removed. If you do a good job, congratulations, like you have a good draft of this matrix B. If you don't do a good job, never mind, this system will not work in this case. Okay? Now, this puts some sort of constraints on the kind of data that you have to see. And in particular, we need to have widespread enough coexistence to be able to find all these coefficients of B, right? Because what we're saying is, to solve this system of equations, for example, we cannot have fewer rows than we have coefficients to find in B, right? Because otherwise we have infinitely many solutions and we're out of luck, right? So to be able to estimate these coefficients for a certain species, it means that the species must appear in at least n experiments, right? If it doesn't, and by n experiments, I mean n distinct communities, not even like if I put another value of like 1/3, 0, 0, that's not going to help, right? So it has to be appearing in at least n distinct communities. Moreover, to be able to, this gets a little more mathematical, but to be able to estimate all the coefficients, you need that each pair of species must appear in at least one community. If these two conditions that are fairly strict are not respected, then you cannot estimate all the coefficients of B. And this afternoon, Abby is going to show you a way to go around this problem, right? But this is a good framework for things where widespread coexistence, right? For example, of these 127 species, like combinations of species that they have for like these duckweed, all of them coexisted. So then we would have plenty of data. Right? In that case, we can also have plenty of data to try out of it, right? Because we have so much data that we can get about the same B by taking subsets and then testing it on the other side.
On the constraints that you mentioned, should each species be in n sets or k sets?
Each one in n sets.
But we can still choose a submatrix B, right? That is of size k.
Yes, but like we need to find, you can see here, right? We need to find these three coefficients. These are unknowns typically. So for finding three unknowns, if I have linear equations, how many equations do I need? Three independent equations, right? So these matrix has to have a rank 3, which means that it has at least 3 rows. Because if a matrix has only 2 rows, it cannot have rank 3.
And the way we choose the submatrix B while removing data of species, is that at random, like in the
Yeah, we can do it systematically. We can take out each community in turn, fit the model without this community and seeing. Like this would be leave one out, cross-validation. That's how you would call it.
So we form the B, take the inverse, multiply it with the I, and see if X is all positive.
See if X matches our experimental data. Yeah. All right. So this is how we would go about doing these experiments of validating our approach. If we had the matrix B, what remains to be seen is how do we get the matrix B? And so there are different approaches. There is a very naive way to do it, which would be I take all my data, I form this equilibria, right? These matrix that I called E of I, right? The matrix with all the equilibria in which, or all the points in which species I is present. And then I take this pseudo inverse, right? So I do this thing, E transpose I times a vector of ones. This is approximately BI, right? So that's one option. This is a very naive way to do it. Of course, it will give you some numbers. I would not trust them very much because what we're doing is really we're putting, like when you're doing this linear regression, right? You're saying AX is equal to B plus some error. So we're putting the error here, right? So we're saying this number on the right are not exactly 1. Where is our error? In our case, it's in the matrix. It's not here, right? So that's why this is a bit of a naive approach that will not give you very good results. But we can use an even simpler results, right? That we can say, well, we propose a matrix B, however we want, and then we score it, right? For example, we say, are my predictions for the mean close enough to the observations? right? And I score this distance between the predicted and observed however I want. And then I can change each coefficient of V of a little bit and say, do I go up in my score or do I go down? And then I try to go up until I cannot go up anymore. I reach like my maximum estimate, like best possible estimate of V, right? So this is computationally very expensive, of course, if you have many species. But if you have 3 or 7 species, doesn't matter. Like you just need to invert a lot of matrices, but computers can do this for you, right? So you don't have to do it by hand. So then it remains to be seen, how are we gonna score these matrices, right? So for each matrix, we can compute using this formula. We can compute what is our expected mean for species I in community K for all the possible combinations that we've seen, right? So imagine that for each one matrix B, we can basically find these two numbers. What is the observed density of species I in community K replicate R? And what is my predicted, given my matrix B, what is my predicted value of I in community K? A very simple approach would be, I take the deviations between these two numbers, right? Square them, sum across all the species, all the communities, all the replicates. I have a sum of squared deviations, and I try to minimize that, right? So each matrix B will give you some score of the sum of squares, and I try to minimize it, right? That is what you do basically linear regressions, for example. Now, is this good? Is this not good? It depends, right? Like if all the species had about the same density, right, in all the communities, then this is not a bad approach. When, however, species change in order of magnitude, for example, as in the data that Sarah was showing us, if we're just taking the difference and squaring it, then a small difference in percentage, say a 1% error in a very large number, something that is 1,000 times larger than the next, is very important compared to a 50% error in something very small, right? So if we're doing sum of squares, we're counting highly abundant species count for a lot more. We want to get them more precisely than the low abundant species. If we want to weight each species, it's dear to my heart equally, so I don't care whether they're abundant or not, what should we do? Right? This is not the first time people had this problem. In statistics, there is like a whole like literature on weighted least squares, right? So now instead of doing this, we weight the deviations by their variance, right? And so if the variance of large things, it's going to be larger, right? Because they even if they vary of the same proportion, the number will be larger. So if we divide them, then we put all the species on equal footing. And so now we're trying to fit equally well the highly abundant species and the low abundant species. And for simplicity, you can take, we don't really know what is the variance, but we can take the observed variance, right, for an estimate. Or even more interestingly, we can say, well, I believe that this distribution is some distribution that I know of. For example, it's a gamma distribution. It's a log normal distribution. It's whatever distribution you want, possibly a distribution that has support over the positive real axis, right? Like the normal distribution would not, right? Like, so if you have, if I say I'm sampling my numbers from a normal distribution, there is a probability of sampling -5 zebras. No one has seen -5 zebras, right? So maybe a distribution that has support over the positive orthant. For example, as I said, gamma log normal would work well. And so now what I could do is, instead of computing the deviations, I could measure the density of this distribution at the predicted parameters, right, or at the observed values, right? What is the density of this distribution given my parameters at the observed values, right? And then you would get a bunch of densities, take the log of that, sum those. So what you would get, then it's a log likelihood that in this case you want to maximize, right? You want to have like a peak around the observed values. So you want to maximize the density around this. Okay, so when we're going to do the tutorial, we're going to try with all these three approaches for this data set on duckweed. And then if you have other data sets, it's very easy to try with your own data sets. All you need to do is to build a matrix like this matrix E that we had. And then we're going to do. Just like to, let me just tell you a little bit about the references, and then maybe we can recap what we just saw, because I went a little quickly. So this statistical model actually has been discovered at least 4 or 5 times that I know of, probably many more, right? Because it's as I said, it's a very simple, very basic idea. So you find it in all these papers. Of course, I'm most familiar with this one because I wrote it, but you can find it like pretty much in the same way in other things. It's interesting that they appear either before or after this other paper. For the tutorial, we're actually gonna follow quite closely the approach that is in the paper by Abby here, that was published in Methods in Ecology and Evolution. It comes with code, so we're going to use some sort of slightly modified, massaged way for this. And then I just wanted to say that the same approach you can actually use to answer other questions. So recently we were interested in these experiments that people do with plants. Can you detect a signal of phylogeny, right? Of that plants that are closely related interact more strongly than those that are less related. And so we analyze data using a minor modification of this setting. Okay, but what I just wanted to stress is just like we have experiments and we observe data that looks something like this, right? This data, for example, on the duckweed, etc., looks something like this when we're looking at only one point, the final point of the dynamics. And now we want to build a set of rules such that all of these observations are consistent with each other, right? We don't want only to use the pairs and now I predict the 6 at a time. If I had the data on 3, 4, 5, why should I only look at pairs and try to predict something that is 6 at a time, throwing away put it in the same foot. 2 main assumptions. The first assumption is if I do the same experiment twice, I get twice about the same result. If that's not, your system does not abide by this rule, this framework is out of the window, right? Like you cannot apply it. Second, we said, well, for simplicity and to reduce the number of parameters dramatically, I'm going to connect all the values of a species in one experiment. It has to be somewhat related to a similar experiment. If I have species A, B, C, D, and now I do A, B, C, D, E, this is not a completely entirely new thing, right? They must be related in some way. And we use this very simple idea of having things linearly related to each other. If you're making these 2 assumptions, you end up with what? With our friend Lotka-Volterra, which, as we said, in a matrix can encode 2 to the n possible communities, which is exactly the type of data that we observe. Okay? And Abby will tell you more, and we will do this in practice this afternoon. Questions? Yes, Sergey.
It's kind of a little bit above my pay grade what you were just telling me, but I wanted to see how it connects to this compressive sensing approach, which James O'Dwyer and others developed. For my understanding is that the compressive sensing lets you do only a subset of those 2 to the power n minus 1 experiments to successfully predict others. Mm-hmm. And you can do it in a smart way in order not to run into a problem of ABCD and ABCDE being roughly the same experiment. So you shouldn't really waste your money on making it.
Yes.
So could you just connect the dots to me, because they're right now in different folders in my brain.
Yes. So this is an approach that can do the same, right? I've seen a bunch of experiments, say I've seen 20 experiments, and now I predict the remaining 54 experiments, right? So it's in the same category in that sense. And it is true that this tells you about experimental design, right? Like which experiment should I do if I can choose, right? And I like this analogy with linear regressions, right? Imagine that I have a set of possible experiments, right, where I have my value of X and my value of Y, right? Like, this is, in fact, like, imagine that these are my communities, right? I can choose different communities, and I want to look at what is the density of species I in community K replicate R, right, for this species, right? So imagine that I can choose. Now, if these species, let's say, are competing, right? So when there's few species, right, imagine that there's only these species, then I will get points here, right? These are my carrying capacity with some noise. When I have very many species, I would expect to be lower, right? Because things are competing, and in the middle, I would be somewhat in the middle, right? So imagine that now I can choose my experimental design. Should I do only 1, 2, and 3? Or should I do the large number, or should I do the small number? So if I only try to estimate, say, the slope of this thing using all these small numbers, then what I have is that I'm making big mistakes when I do the large numbers, right? Similarly, if I only experiment with things where this thing is very low, I would make mistakes in this way. What is the best solution? Is to mix and match, right? I take a few experiments from here, a few experiments from here. I anchor this plane because this is basically a hyperplane, right? I'm saying pick all the numbers such that this is a hyperplane, and then I get a much better estimate. So in the paper with Dan Maynard, the paper in Nature, Ecology, and Evolution that I was referencing, there's a whole discussion of how do you go about doing a smart experimental design. Similarly, you can do this in a Bayesian framework. So imagine that now I ran a bunch of experiments, and now for each Bij, I could say, what is my uncertainty, right? So then I can choose experiment to reduce the uncertainty, right? So I can choose in a smart way to what is the next experiment I should do.
So you do iteratively experiments, not plan them ahead of time.
Instead of doing all the experiments in one go, you could do, I do first 20 experiments, I get a draft, and I say, oh, these numbers I already pinned down very clearly. These other ones I have uncertainty. What would be a good experiment to do to pin down these other numbers? Right, so this opens like a lot of good questions about experimental design.
And a compression sensing, again.
I have no idea what the compression sensing is.
Okay, well,
so you will have to explain it to me. Yes.
So this approach allows us to be able to say that the Lotka-Volterra approximation, in which basically there is pairwise interaction assumed, is failing, and that there is there might be some intrinsic three-body terms that we need to write down and so on, things like that.
Absolutely, that's a very, very good point. I want to make maybe a slight distinction, right? When we're talking about is the Lotka-Volterra working, we could think about is Lotka-Volterra reproducing trajectories. You've seen like today these very complicated trajectories. I can bet $1,000 that Lotka-Volterra will not reproduce those trajectories at all, right? So then you're like, but you just showed me Lotka-Volterra, why would you do that? Here, we're not looking at trajectories, right? Here we have a set of points, right, that you can think are making some sort of like a hypercube, right? Like we have a species 1 by itself, species 2 by itself, right? A species 1 and species 2, right? And so we have all these 2 to the n points in this n-dimensional space, right? And what I'm asking is there a matrix which, when I do this kind of a calculation, gives me points that are closer here? Okay? So if it does, it doesn't mean that the trajectories would be followed by Lotka-Volterra, but it tells you the places where these systems go, these, say, equilibria, there would be a Lotka-Volterra model that has the same equilibrium, which is a much easier thing to match than matching the trajectories. But then, what if it fails? If it fails, it's exactly what you're saying. It's either that there are higher order terms that I'm completely neglecting, so I'm failing, or either the first assumption, I do the same experiment, goes to different places, Lotka-Volterra cannot do that, and this system cannot do that either. Second, where I go depends not only on the pairs, but on triplets or higher order terms, or like powers of the densities, non-linearities that are not included. Again, if this thing fails, it tells you that, if you don't include these other terms, you're not going to be able to fit your data. So this would be a failure of the statistical framework. So that's, I think it's something that it's good, is you have something that is fairly clean cut, such that you can say, it failed. If it failed, it means my system does not abide by these 2 assumptions that we made. So you either make other assumptions, or you give it up and you don't model your data. And all right, I think it's lunchtime, but we can discuss more about this this afternoon in the tutorial. Thank you.